This is an overview of an article titled "A Screening Approach to Identify Clinically Actionable Variants Causing Congenital Heart Disease in Exome Data," published in the March 2018 issue of Circulation, Genomic and Precision Medicine. Congenital heart disease, or CHD, is the most common type of developmental defect, affecting about 8 in 1,000 live births, or 1.35 million infants each year worldwide. Causative genetic abnormalities, either inherited or sporadic, can currently be identified in about 30% of cases. Single gene mutations can be causative for both syndromic and isolated CHD, but some cases are believed to be defined by complex multigenic and environmental interactions. The authors of the present study utilized whole exome sequencing data from 30 families to identify disease-causal variants for congenital heart disease. 10% of families displayed pathogenic variants in known CHD genes, namely PTPN11, TBX5, and TFAP2B. Another 33% of families carried rare and damaging variants, segregating with disease in genes with supportive evidence in the literature. In the remaining affected families, no causal gene variants could be identified. The cohort of the study consisted of 30 unrelated families, including individuals with a family history of CHD, as well as sporadic cases. Heart defects were confirmed by echocardiography. In all families, at least a trio, consisting of the affected proband and their parents, was analyzed. To see a table with detailed clinical characteristics of the study participants, please refer to the published article on the journal website. The author's strategy to identify causal variants for CHD from exome sequencing data was a two-tiered approach. First, the authors generated a list of high-confidence genes causative for CHD by querying multiple databases, combined with stringent filtering criteria. A detailed description of the process can be found in the supplemental methods of the published article. This high-confidence CHD gene list will continue to be updated after publication to account for the growing number of published data about human gene-disease relationships. The most up-to-date version is available at the web address shown here. Screening of affected cases for predicted damaging variants in these 90 high-confidence genes identified 7 variants that fulfilled the following criteria. 1, they were rare, with a minor allele frequency of less than 1% in the Exome Aggregation Consortium database, or EXAC, and the 1,000 Genomes database. 2, they were predicted to be damaging based on 3 different algorithms. 3, they were supported by evidence in the literature. Of these 7 variants, only those in 3 genes were rated as pathogenic according to the guidelines for the interpretation of sequence variants jointly published by the American College of Medical Genetics and Genomics and the Association for Molecular Pathology in 2015. More details on the variants and genes shown in this table are available in the published article. In the second approach, exome sequencing data from every family underwent a comprehensive screen to identify potential disease-causing variants in an unbiased way. Variants in any gene were filtered for being rare, predicted to be damaging, and having supportive evidence in the literature. They also needed to segregate with disease. All possible Mendelian inheritance models were tested. Here are the results of the comprehensive screen. More details on the variants and genes shown in this table are available in the published article. In conclusion, with their two-tiered approach, the authors identified disease-causal variants in known CHD genes in 10% of the cases and additional likely causal variants in another 33% of the cases in a cohort of 30 families with sporadic or familial CHD. From a clinical perspective, this study addressed an unmet need to improve the genetic diagnosis of CHD by generating a dynamic, high-confidence list of disease-causing genes. Furthermore, the authors demonstrated that screening variants from whole exome sequencing using a well-defined framework can identify additional likely causative variants in additional genes. Thus, the employed two-tiered approach provides a means of screening high-confidence genes as well as identifying additional likely disease-causal genes to improve the molecular diagnosis of CHD.